Hey, and welcome back to lecture 6.2, where we'll be talking about membrane transport and bulk transport, which is a type of membrane transport. Anyway, lecture question 6.1, of course, you're going to want to know the answer. If you said the answer was C, you'd be correct. That was a fairly straightforward question, but you're going to see some of those pretty straightforward ones on the exam as well. So, all right. Anyway, back to the content for today, specifically membrane transport and bulk transport. Okay. So let's go into specifics about, I kind of left it off, a general introduction to what transport is, what passive and active transport is. So let's talk a bit more specifics of it now, right? Starting with passive transport. So there are kind of three different kinds of passive transport: simple diffusion, osmosis, and facilitated diffusion, which uses proteins. We'll get to that. Now, the things being moved, right, are moving down a concentration gradient. In other words, from high concentration to low concentration. Also, ATP is not required. Now remember, ATP is pretty much synonymous with energy in a cell, so you don't need to invest energy to make these things happen. They can pretty much just happen immediately. These things will also keep happening until you reach a dynamic equilibrium, which means there's still going to be movement back and forth, but the net movement is about zero, right? So like you're still gonna have like two going this way and two going that way, but it's gonna be about equal, right? All the levels are gonna even out. So let's go through one by one, starting with diffusion. Diffusion is the tendency for molecules of a substance to fill the available space. For example, if you have a bottle with ammonium gas and you open it up, the gas is going to leave the bottle and fill the empty room, right? It's going to go from a high concentration in that little bottle to the very low concentration in the room. But hold up though. So that's how diffusion works in general. We're talking about cells, right? So how does it apply to cells? Well, if there's a high concentration of a substance outside a cell and a lower concentration inside the cell, then you'd expect some diffusion, right? But what do we have to deal with to actually get into that cell? What's stopping? What's separating the inside from the outside? Yeah, the plasma membrane that we just talked about in 6.1. And what we know is that plasma membrane is made up of fairly tightly packed phospholipid bilayers, which have very hydrophobic, nonpolar insides. This means that some smaller and nonpolar molecules like oxygen gas or carbon dioxide or even nitrogen gas can actually slip through without being stopped by that very hydrophobic inside of the phospholipid bilayer. Now, even some slightly larger nonpolar molecules like hydrocarbons can actually squeeze past those hydrophobic tails, and some polar molecules can too, actually, as long as they're not too big. In other words, as long as they're very small and they don't have a charge. It's the other important thing, right? So they can be polar, but they have to be small and they can't have a charge. So water is a really great example of one of these small, uncharged polar molecules that can slip through the phospholipid bilayer. In fact, we have a specific term for that process: osmosis. Okay, now this is where we get a little tricky though, because we've been talking about high to low concentrations, right? For example, it was a high concentration of ammonia gas in the bottle to a low concentration outside the bottle. Right. Well, with osmosis, that means we're specifically referring to high to low concentrations of water, the solvent. Let's use a really simple example here, right? These dots. So we have blue dots, which will have represent water, and red dots, which will be the stuff that's dissolved in the water, the solute. Okay. So in this particular scenario that we see here, we'd expect what to happen. What do you think? So we have 3 blue dots on one side, 1 blue dot on the other. What would you expect to happen? Right. So the water is going to move from a higher concentration to the lower concentration. Pretty straightforward, right? But this is where it gets a little complicated with osmosis. So it could also mean that you're moving from a low to high concentration of the solute, the stuff that's dissolved in the water. Meaning you got a little bit of a solute on one side and a lot on the other. Your water is going to move, so there's actually a relatively similar quantities or relative quantities of water and solute on both sides. It's a lot of words, a little confusing. Let's take our simple dot scenario here, right? We have one cell with 2 dots of water and 1 dot of solute. We have another cell adjacent to it with 2 dots of water and 3 dots of the solute. Now the water, you have the same amount of water on both sides of the membrane, right? 2 dots. However, the right side here has a lot more solute. So what's going to happen? You're going to have water move from the left to the right to achieve similar ratios of water to solute. So blue dots to red dots. So they're kind of equalize. It's going to try to equalize, okay? Okay. Now I keep saying solvents and solutes, and that can be a little confusing. So, I mean, one, they sound really similar, obviously, right? But so let's go over these definitions real fast. So solvents are substances capable of dissolving other substances. Water, for example, is an almost universal solvent, doing a great job at dissolving pretty much anything polar, specifically, okay? A solute is the stuff that's actually being dissolved by the solvent. So lemonade powder is a pretty straightforward example, right? Dump your powder into water, mix it up, dissolves, now you got lemonade. What's important to remember, though, is that water always moves towards the higher concentration of solute. In other words, when it comes to osmosis, we're always talking about the water moving, not the solute moving, okay? Now, this leads us to the concept of osmotic tonicity, or osmosis tonicity. Anyway, which is basically the ability of a solution to gain or lose water by how much relative solute it contains. It's a little confusing, but maybe we can work through scenarios. So a cell, for example, can be isotonic, where the solution inside the cell can be isotonic, where the solute outside the cell and the solute inside the cell are at equal relative levels, right? So this makes sense. Iso comes from the idea, the root of same. So iso equals same. So same levels of solute inside and outside of the cell. Isotonic. So there's going to be no net movement of water, right? There might be still some exchange of water, that dynamic equilibrium thing, but there's not going to be any like all the water from here is going to here, right? Okay. Now, hypertonic solutions is when there are more solutes outside the cell than inside the cell. You'd actually expect a cell to shrink as the water moves from the high concentration of water inside to the low concentration of water outside. Okay? So all the water is going from this not concentrated solution inside the cell to the very concentrated solution outside the cell, and it's going to shrivel the cell. Now, this is opposed to hypotonic solution, when there's less solute outside than inside. This causes the water to move into the cell to try to reach a relative equal concentration. This can result in the cell actually swelling or maybe even bursting if too much water rushes in. So I kind of try to keep this straight by remembering or thinking of hippos or hypos, because the hypotonic solution or hypotonic solution? Hypotonic sounds way more fun. Anyway, hippos are very large and round, like a hypo or hypotonic solution cell, right? Okay. Now remember, it's important. These are all relative terms. So what might be a hypotonic solution in a cell that's hypotonic in, say, freshwater solution might be hypertonic in a saltwater solution. Make sense? Okay. All right. So that's all this like kind of passive stuff, kind of moving in and out, kind of just diffusing across the barriers, right? But what about the stuff that can't actually get through those membranes? Maybe because the molecules are too big, or because they have a positively charged, like a hydrogen ion or a calcium or sodium ion. Well, you can actually use one of those transmembrane proteins that we talked about in lecture 6.1 to help things along. So facilitated diffusion does just that. So there are proteins that are designed to kind of shepherd specific molecules through the phospholipid. Now, some of these proteins are called channel proteins, aptly named because they have a special channel that goes from one side of the membrane to the other that kind of specific molecules can actually pass through, right? Now there's other proteins that act as carrier proteins, almost like they're kind of picking up the molecule and then passing it through to the other side. Now, this functions by the molecule that binds to the protein causes the protein to actually change its shape. And that shape change is what brings the molecule to the other side. Now, in both of these cases, the reason it's considered facilitated diffusion is because there's no energy that's being utilized to actually transport these molecules. It's simply a product of how these molecules interact with the proteins and change the shape or move through the channel, right? So also important to keep in mind here, facilitated diffusion follows the same principles as other forms of passive transport, in that things are only being moved from high concentrations to low concentrations. Okay.